So Dr. Bill Campbell posted this research study on his Instagram the other day, and I learned a lot from this, so I wanted to share it with you guys. So this study looked at 6 untrained individuals and wanted to investigate the rates of muscle protein synthesis, or how the muscle grew in response to amino acid availability. So if we look at the y-axis, we have muscle protein synthesis. On the x-axis, we have time. So what they did in the study, they infused a steady state or a steady concentration of amino acids via IV into the subject's blood, and they measured the rate of muscle protein synthesis compared to baseline, with baseline being 1 time. So as you see here, there's a rapid spike at about 2 hours where muscle protein synthesis reached about 2.8 times the maximum or the baseline value. But then thereafter, even though amino acids were steadily infused into the blood, there was a rapid drop. So this study tells us a few different things that can be summarized with this little figure here. So even in states where we may have excess protein, say if you have a protein shake, a steak, and something else that's really high in protein, all those proteins are going to be digested and metabolized into amino acids, and they can go a few different routes. One, if there's too much, it can get converted to urea, which is just a metabolic waste product of proteins, and then urea eventually gets excreted in our urine via the kidneys. The excess amino acids can also be used for immediate fuel source, which just can be converted into ATP, which fuels things like, skeletal muscle contraction, things of that sort. Or it can be converted into storage molecules, because amino acids are gluconeogenic, meaning they can form glucose, and they can be stored as both glycogen or fat. Another thing that was pretty interesting was that muscle protein synthesis response was pretty analogous to the insulin response, which was pretty interesting to me because I always was told you should have something high in sugar after you work out because it leads to an insulin spike. When your insulin's high, it allows you to uptake glucose for glycogen repletion, and it also allows you to take up amino acids. But I learned through this study that amino acids themselves are gluconeogenic, as seen in this study, because there was also a 3-fold increase in insulin after the infusion of amino acids. Also, if you were to take this figure and elongate the time out over a longer period, the researchers also found that plasma amino acid levels have to come back down to baseline in order to achieve this spike again. So what does that mean? That means that if we take in excess protein, according to this study, it can be partially detrimental to the rates of muscle protein synthesis. So you might be hurting yourself by taking in excess protein in one sitting as opposed to divvying it out over a few different meals. And that idea is known as the refractory response. So they hypothesize, they obviously don't know the whole mechanisms behind this, but they're hypothesizing that there are certain things within our DNA that are able to sense when there is excess energy available, and it stops the rate of muscle protein synthesis. And that makes sense, right? Because although it might be cool to be huge and be really jacked and get muscles, physiologically speaking, that doesn't make too much sense, since if you're increasing your mass, you're going to increase the amount of energy needed to fuel the body. So it wouldn't make sense to continually have spiked muscle protein synthesis at all times. So technically speaking, if you're going to put this information to use, they suggest that having a meal of 30 to 35 grams of protein every 3 to 4 hours is what allows you to achieve this spike of muscle protein synthesis, while also allowing plasma amino acid levels to return back to baseline so that this spike can be achieved again. And again, there are limitations to the study. It was only 6 untrained subjects, which is a small sample size. And it also can be, a lot of these processes can be very different in trained subjects. We know that there's a ton of different genes that are turned on by people who are physically active. So that's another limitation that we can assume in this study.